So, good morning, everybody. I'm happy to see real faces again. So today we are going to start with the quantitative big imaging, images to statistics, and I'm Anders Kästner. I try now to do combined, so I have Zoom running. I will also record the videos, and so there are many options in case you can't attend on site. So let's hope it works well. And it's off. There it is. No, oh no, it doesn't work. Okay. Zoom is interfering with my stuff. Okay, in that case, I'll just go on with normal space. So today's lecture, first about the course, what we are going to do, motivation to use quantitative methods in imaging, and then a little bit basics about what are images, where do they come from, and also some basic science theory and looking into workflows, how to work with images. So I'm going to do these lectures based on Jupyter notebooks. So what you see here now is some lines of code that I need to run the notebook, and they are usually in the beginning of every notebook. And I need to execute it in order to continue working with this notebook. Sometimes there is a little bit code on the slides, but mostly I try to hide it. But in those cases when I think it makes sense to show the code, you will see it. So first starting up with something about the course, who am I, who are you, etc. And also a little bit about why we have this course at all. So first of all, I'm Anders Kästner. I'm a beamline scientist at PSI, working with neutron imaging, and been there since 2008. Now I'm also teaching here at ETH. And before that, I was working as algorithm developer at Varian Medical System. And I was a PhD postdoc at Institute for Terrestrial Ecology in the soil physics group, mainly doing images and image analysis for soil. And originally I got my PhD in Sweden at Chalmers Institute of Technology, and topic was signal processing. So that's a little bit about me. I was always fascinated by images and being able to tell something for images. So with my background in computer science, I always wanted to tell the computer how to interpret images. And I think that's quite challenging because as human, you can always look at images and see, well, that's that item, and that's that one, that's maybe that one, and so on. And now the challenge is actually to convince the computer to make the same conclusions. And that is a challenge I like. Then I also have as support Stefano. He is a PhD student in the X-ray microscopy group at also at PSI and partly here at ETH. He's my teaching assistant, but right now he is doing an exchange scientist program, so he's not on site the first 5 weeks. But he will be available in the background. He's actually logged in right now. And for those who are doing the Zoom approach, he will also keep answering questions if there come questions in the chat. He will also help you during the exercises, and the best way is actually contact him in, making an appointment, and then you can have a Zoom session with him directly, because we noticed the previous years that Zoom sessions usually tend that we sit there waiting, possibly there is something, and it's much easier if you just make an appointment with him if you want to or me actually also, if you want to have something cleared out from the exercises. For that, we also have Slack as communication background, as communication. Oh, that one didn't work. Okay, for you on Zoom, the screen went black probably. I'm showing now the user interface of Slack, which is a chatting platform, mainly used, a lot used for software developers. And it's a platform where you can type in your questions. Both Stefano and I will look at those chats and answer questions. You can also ask questions among yourself, etc. So that's a little bit about communication. Of course, our email addresses, they are also on the slides if you want to look at those. So a little bit about the background. I already saw on the list of participants that there is a wide variety in background. We have had art history researchers, we have had chemists, mechanical engineers, biomedical engineers, computer scientists, mathematicians even, physicists, and so on. So it's a very wide group of people that are around here. And the skills, what you know about image processing, programming, what kind of math you have approached before, varies very widely with that. So we have experience from little baby till the advanced climber. We have data of different types, from very simple objects till complicated structures that needs to be understood in some way. Also, the way how you work varies from you prefer to point and click and draw things to some of you may even are skilled programmers already. So it's a very wide clientele, and would actually be interesting to know what background you have and to know you a little bit better. So maybe during the exercises, maybe you can tell something about what you expect and what your background is. So I would appreciate that actually. So how will this work with this very heterogeneous group? So first of all, the assignments in the exercises, they are built up in a way that it's more like a recipe describing. You have some ideas, you have some concept, and then you have to solve some problem with a few lines of code. Usually it's set up in a way that you don't have to program too much. Maybe towards the end, there will be a little bit more complicated tasks, but first of all, we start softly. So what we have will be one lecture per week or lecture set. So that's the 2 hours now. And then we have also the exercises, and they're not mandatory to pass the class, but it's pretty good to do them to understand more how to work with the data. In particular, those of you who actually have their own projects, it makes well sense to really touch and try and see how it works. Maybe even grab in your own images and see how it works. So the exercises are mainly done as Jupyter notebooks, and in the exercise session, I will first help you to get started, getting those exercises. If you don't have Anaconda installed yet, someone wants to say something. Do I hear right? No? Okay. Someone is not muted, maybe. And then we have the project. It's a small project where you can take everything, put it together in one basket and try to analyze images and use the knowledge you have gained during these classes. It can be some project we propose, but it can also be part of your master project, can be part of your PhD project. And with that, you would also gain a little bit of our support getting further on in your own analysis. And that will be in the end that you will present. Excuse me? Someone on Zoom is trying to talk, or is it just unmuted? Oh, probably just unmuted. And in the end, last lecture, you will present your results, and everybody can also gain from your experiences. Yeah. This is the overview of the course. You probably already saw some of it on the web page. It's just a more compact form with less graphics in it. But in principle, it's one each week. There are 2 weeks when there are no lectures. So that's for Easter and Ascension in April and May. But otherwise, there will be a lecture every week on Thursdays this time in this room and on Zoom. Then there is, as always, a lot to read. If you want to get deeper into different topics, here are some short articles about image processing and how to work with it. Reproducibility is quite important task today, or repeatability. And there are some nice papers here to look into. A book that's a good introduction to image processing in general is the one called Image Processing Handbook by John Russ. It is available online, and don't buy it. It's about, I think, 1,500 pages or something. It's quite heavy to carry in the bag. So anyway, it's online, so you can download it. Another one is Morphometry with R by Jean-Claude, and it's also available online. Has the disadvantage, it has a lot of good theory in it, but the disadvantage for my point of view is that it's using R instead of Python as language, but the knowledge in there is worth taking a look at. So, motivation. Why do you take this class at all? So usually you have done some imaging experiment, you got your proposal, yay, I can do experiment. Go to a beamline or go in a lab, doing the experiment, produce a lot of data, sometimes loads of hard drives of data. And then you come home, look at your hard drives, no idea what to do. And that is the gap that I'm hoping to fill with this class. And it can be a lot of data in the beginning. It's more about having a workflow, or maybe even being able to plan, how can I do the experiment in the best way in order to easily analyze it. Sometimes just depending on how the order you do things can make the analysis easier or harder. So it's the point where I come in here, how to analyze and work with the data. And the motivation that we want to do it is we want to understand our data. So in the beginning, you have, in this graph, you have done some acquisition, you have data, then you do reduction, normalization, removing some artifacts, etc. Maybe you do a tomography reconstruction. It's still just data in the point of view for a computer. Then you do some filtering, still only data. Then we come to segmentation. Then we start adding a little bit of knowledge about our data. We have separated into different classes. And then comes labeling. Then we have identified, oh yeah, there are items around in the image. Going on, starting doing measurements on the items. And then we start getting more knowledge. And we get a table with values. and it's getting better. From those, we can start categorizing, classifying the groups in a table, and that will then give you even more knowledge. And in the end, you can do some statistical inference telling, yeah, this group has these features, this group has those, they correspond to our hypothesis, they don't, etc., etc. And then you're getting closer to what you want to publish. And then we are rather talking about knowledge than data. And the procedure, you can set up some kind of funny algorithm. You have done your images, get slices, then you do some fuzzy algorithm with fancy stuff and magic stuff and some math stuff and complicated stuff, etc., etc. That's not really the language we want to talk about. But in the end, maybe you come to the point that, well, science says ice cream is good for bones, maybe. But we want to get away from this fancy magic stuff and really describe what we are doing. Because no referee in a journal would look good at something that we say, you do fancy stuff. And the question is how to do this for one image. Then it's pretty easy. Just click around a little bit and yay, you got some results. But what happens if you have 10 images, 100, 1,000? Who knows? You may get the result, but the question is how you can do it efficiently and work with it in a good way. So you have the experiment lifecycle where you first design your experiment. You have to find the right technique, the right setup, etc. Preparing the samples. Then you do your measurements. And that's usually a pretty short time of the whole lifetime. Then comes management. When you have a lot of data, you have to plan how to manage your data. If you want to store it on hard drives, you want to store it on a cloud archive or something like that. And all this copying and overhead needs to also be well planned in order to get reproducible data. And today, it's even more important to have public data, open data. And then it's important that you can have well-described procedures where you can find and how you can use the data. And finally, the part that actually takes a lot of time is the post-processing to do everything I showed in this graph before. And looking over the years, sorry for the very small text. In 2000, you had experiment was one part. Then you have some measurements, experiment planning. Then you have the measurements, which took a long time. We had less good infrastructure for doing the experiments. Then the management, well, it took some time, of course. And then you came to doing the post-processing. And over the years, you produce the experiment design time relatively takes much less compared to the data management, and the measurement, you barely see it because the beamlines, they can produce terabytes of data in short time. And that makes it, the experiment part itself, relatively small compared to the management and the post-processing. And when you're talking about copying a terabyte of data, it actually takes time to get it from one point to the other, in particular if you have to do it over a network, and if you have to do it a couple of times to get it from a cluster to another, to get it home, then this time takes a lot. So we have very high, I have already mentioned that we can produce a lot of data very fast, and the detectors, they're getting bigger, they're getting faster all the time. And today, we can get a little bit more than 2K by 2K images at a frame rate of 1,500 frames per second. That is 8 gigabytes per second. And different software, they are saturated with this data rate within 60 seconds. So you need to find a way to handle this flow of data in a good way. Just to compare, a single camera produces more information per day than Facebook. And that's pretty impressive, actually. You can click onto that link and see that. That's actually a couple of years ago, so I don't know if it's still valid, but anyway, it's quite impressive that images or pictures that are uploaded to Facebook is less than these cameras can do. And it's also 3 times as many images per second as is uploaded on Instagram. Then we have different sources of images. We have X-ray images that produce data rates up to 10 terabytes a day. We have optical imaging, which produces 500 megabytes per second in extreme cases. But that needs to be reduced pretty quickly at source, otherwise you will not have a chance to handle it. And then if you go to personal devices, a GoPro produces 60 megabytes per second. That is almost 4K by 2K, 30 frames per second. And that's a pretty cheap device, and it still can produce also high amounts of data. So how to handle all these images. And the thing is, as soon as you start to process the data, you create more data. It's kind of a law of nature. As soon so you have your original data, then you do some filtering, then you have 2 copies. Then you do some next step, then you have yet another copy. So looking at another graph, somewhat similar to the one before, then you can see here that with many steps here, you have linear increase. Every time you do an operation, you plus on the same image size again. Because we're talking in this region here, we are talking about the full volume is processed and moved from step to step. Then when you come to segmentation, then something happens, because then you reduce the amount of data in each pixel. You still have the full volume, but you go from maybe 32-bit data down to 8-bit, and that's already a reduction. Labeling is about the same, depends on how many items you have. Maybe you can work with 8-bit, maybe you have to go to 16-bit, I don't know. But anyway, there is also some increase. But when you start doing measurements and classification inference, then you work on tables. And that's extremely reduced data amounts. And then you can see also then this curve, it flattens out. So how much is a terabyte? Let's start. You have an image, 1K by 1K. That's 1 megapixels. I don't say how many bytes per pixel, but let's say it's an 8-bit image. So if you look at a 1K image every second, how long would it take you? So let's do a calculation. So we have 1 terabyte is this amount. Then we have image size. And this is metric, by the way, but anyway, in bytes. So this is then 16-bit. And an hour is 60 by 60 seconds. Then we do the calculations. And you can see down here, that it's about 140 hours to view 1K image per second for 1 terabyte. So you can't do it. It's impossible. So you have to let the computer do it. The computer can do it a lot quicker, even though it also will take its time. Oops. So let's look at the real image. We have here an image with bone cells in it. And first thing is you want to count how many cells there is in this slice. You can see all the white spots. And some needs to be ignored. They are too large, or they are shaped in a strange way, so they can't be a cell. And you have to decide which are cells and which are not. And then you want to know, are there more cells on the left side and then on the right side? Or are the larger ones, are they to the right, are they larger than the one to the left, top or bottom? You want to make conclusions about how big they are and where they are. So one image, okay, you can sit down, count, exclude, etc. Can maybe also do some qualitative estimates about, yeah, that one is larger and that one is not. It works in one image, but then we start adding it, because when we do science, you want statistics. So we repeat the same thing for 96 samples. And this time it's in 3D. So we have, instead of just one image, we have 2,000 slices. That's getting pretty hard to do by hand. And also, when you move from 2D into 3D, it's also harder to do the segmentation, because on one slice it looks like, nah, that's nothing. And then if you look at the next one, oh, it was something. So it's not so easy to do this analysis in 3D. You can use volume rendering programs to look at it, but still, 3D analysis is not so easy when you have a 2D screen. Then we want great variations in the population. So it's not only one kind of phenomenon you want to study, or you want a control group and you want to have different treatments. And then all of a sudden you have 1,090 samples. The first struggle is to get all these samples scanned. And in this particular experiment, they actually had a robot moving in and out the bones in the CT scanner to do this, because it wasn't possible to handle it manually. So they had a tray with a certain amount of samples put in, and then they set the scanner to work. And then they came back and put in the next tray. And if you have this amount of data, then it's the question how you can analyze it and process it. And then it's even more complicated. You want to have some more quantitative metrics, and it's sometimes easy to see it by eye, but if you want some softer metrics, like how well aligned are these cells, so if they have some anisotropic shape, are they all directed in the same way, or are they just randomly oriented? And on this slide, you have the option kind of aligned. Some are going that direction, maybe. Then there are some going in that direction. And then you have well aligned. They are pretty, these items here, they're pretty straight oriented. And then you can have not really aligned. And that's just chaotic randomness. So, and then it's the question on, is it more aligned in that region than in that region? Or is it, how are the alignment region-wise organized? And that's also a question how you can put numbers on it, because in the end, you want to have numbers that can tell you this information. And to make it even more fun, we can have dynamic information. What happens if you start running an experiment in real time, and this is a foam that is evolving. Actually, I don't know. It could be actually coffee foam that is bubbling up. And then it's a question, how many bubbles are there, first of all? How large are they? How fast are they moving? Do all bubbles move at the same speed? So maybe at the walls, they have a different speed than in the middle. And the other question would be, do the bigger bubbles move faster than small ones, or opposite? And do the bubbles rearrange, or maybe even merge? that's information that you want to know maybe about this sample. So now let's talk a little bit about scientific methods. So what is the purpose of it at all? First of all, you want to discover and validate some new knowledge. That is the main goal. And how we can do it? First, we need to build up a scientific approach to convince people that it actually is something new and you actually did discover something. And fortunately, a lot of people have done stuff before you. So of course, you have to build up on that and then continue from that point. And it is, of course, important to have a qualitative information. But that is, first of all, very difficult to reliably produce and scale in larger data sets. And quantitative information, let's call it more perfect, but still it's far from perfect, because you can always, everything depends on which metrics you use and how you define your analysis. And then it's important to show that your analysis actually proves to be doing that what it's supposed to, also compared to what other people did. And the other side is someone else should be able to reproduce what you're doing. So science and imaging, we want to interpret these images in a good way, but we don't want to add too much programming effort, and at least not in the beginning. It may happen that you need to implement your own new analysis approach, but often there are libraries that can help you to do the analysis, and please use them, because otherwise you will probably spend the whole PhD on just one single problem, and you don't come along with the rest of it. And the problem is that in different physical measurements, it's easy. You can easily say, if you measure the temperature, it's easy. You just put in a thermometer and confirm, okay, it's 50 Kelvin in here. But if you have an image, there are so many factors that you have to take into account. And there's a lot of fuzziness. For one thing, because of noise, because of image sharpness possibly, and variations in the measurements, and that can lead to quite confusing analyses. And in some cases, by choosing the wrong tool, you can actually also produce completely misleading results. And in image processing, there is a lot of algorithms out there. There are even journals, many journals on image processing. There are also many tools, commercial, open source, within your own group even. People who have done their processing in the beginning, so you have something to start with. And there is rarely, or I would say almost never, a case where you can use a single image processing operation that gives you the result. Mostly you have to do a chain. As you saw on my graphs, I had, what was it, about 10 different steps that you have to go through in order to get your results. And each step in itself may be more or less complicated, and may include also their own recipes for how to proceed. And in some cases, you just have to experiment with the images, getting an idea how your data best can be approached. There is, of course, typical template path through the data, but still the details when you come to your experiment conditions may be different. Maybe you need to take that kind of filter instead of that kind of filter because you have those features in the image. Who knows? And that is a little bit what you have to experiment and get experience how to best work with the data. So when you do 3D imaging, or in general, the first thing you have to ask yourself, there are some important questions. What do you really want to get out of your data? And in some cases, say in the early pioneering days of imaging, it was great if you could just show a volume rendering like this gold piece here. But after a while, that wasn't sufficient. And today, it's even publications done with imaging, you don't even see the images. You more see something like that instead. Both these images come from investigations in archaeology. One is just they wanted to see how this piece was manufactured. It's a gold piece dug out from a Viking grave in Sweden. And this is the corrosion of a Roman nail. And they wanted to see how to preserve the, how to best preserve the nail against corrosion and see how salt migrates. And this in particular is even a combined measurement of neutrons and X-rays. So you see that there are really different categories or ways how you can analyze your data. And everything goes down to what you really want to know. So the initial questions you have is, first of all, what am I looking for? Good to know. It's a good point when you go up in the morning, what do I want to know? And then you have to ask what you're looking for. Is that question too specific or is it too general? Like, can you really do an image analysis based on that question, or do you have to reformulate it? And then it's also, then based on that, you set up your experiment. And then when you set up the experiment, how can you optimize the experiment in a way you can get a better analysis? And very important, there are usually two kinds of vocabulary. One, in your own field of research, what topic it may be, physics, archaeology, biology, whatever, use one kind of vocabulary. And then there is a vocabulary, that very technical vocabulary of image analysis. And how can you make a bridge between those? And that is often the hard task, how to convert what you want to know in the terms of image analysis, and in the end, in some kind of code. And it can be that you want to compare different treatments and get differences between them. You want to understand or model a process. So you can see, for example, water uptake in soil. Then you can see that you have some kind of water retention curve. And that curve can actually be measured in the images. But you need to prepare the data in a way that you can do it. And then, of course, it can also be just that you want to inspect components. You want to see sizes or presence of sizes or objects in the image. And it's also very important that if you have a priori information that can guide your analysis, that's great. Use it if you can. Or if, sometimes it's not possible. It's the wrong kind of a priori information, but you usually, when you start designing your experiment, you usually divide it in different treatment classes, etc. And that information is already good to know. And this follows then what is the purpose of the experiment. You want just to do a 3D visualization. Sometimes it's actually sufficient. So like in this gold piece that I showed before, it was sufficient to do a 3D visualization because the rest would be more investigating and see how things appear inside. It can be sample characterization of one or many samples, or you want to model process parameters, and so on. So now comes the question, what can we actually measure in images? Then we divide it into two different classes. One is quantification from the gray levels. So you really see the gradients in gray level. And on the other side, you have quantification from shapes. The first one is about looking into material composition, material transport, etc. And the other one is to identify and count items, measure volumes or dimensions in general, characterizing shape. So if you have donuts and macaroni, for example, whatever those shapes, and you want to see how many you have of each kind. So what you need to look into then is topics like finding hidden objects, measuring, counting, mixing ratio would be for the transport, and also tracking changes. So it could be that you want to see how things move or how things grow, or just how the gray level is changing. And looking at this picture, you probably have seen similar things before. There's an optical illusion. And the question is here, without measuring, which of the center squares is brighter? If you've done this test before, you probably know already they are the same. So in order to know it, you have to actually do measurements in images. So in this case, you would take a region in here, measuring the gray level, region in here, measuring the gray level, and then you would confirm that, okay, it's the same. But in order to do this, we need to do some kind of quantitative analysis and not just saying, yeah, maybe. The other thing is also the problem how to measure intensity gradients. This is another optical illusion. The question here is whether these, it's actually not so good light here now, but if these bars, if they have constant gray level, the optical illusion, the eye actually amplifies, so it looks like it's darker on that side than on that side. And again, if you would take just a line profile across here, then you will see that the profile is actually a pretty nice staircase, and not as you would perceive it, something like this. So again, here it's really important that you do measurements to see that things are actually constant. Oh, just need to clear that. So, and let's see. Now it's a question, should we make a short break? It's 10 o'clock, normally break time. Yeah, let's do some minutes. What is the routine normally? Do you have the 15 minutes break, or you just go on, or how does it work?